Okay, guys. This is chapter 7 of week 8. This is conception and fetal development. The conception and development, I'm sorry, of the embryo and fetus. Um, so I'm going to kind of run through this. There's a lot of A&P we're going to review quickly, and then we're going to review fetal development very quickly. And then at the end of this, I am going to go over some notes with you very quickly about um, I want you to study everything and I want you to learn everything, but I'm going to go over with you quickly some notes I took that pointing out some very important things um, to you that you might want to make sure that when you're studying everything, you really study these very important things. You do have the exam this weekend. I don't remember if I said it in the recording for chapter 6, but um, uh, so I did have to leave town suddenly and had to do these recordings. I felt really bad that you have a test this weekend and I'm not there with you in lecture. So I did make some notes about some things that would be very important for you to focus on in both this week, these two recordings for this week, and then last week's lecture. So I'm going to go over the chapter 7 very quickly. I am available if you have questions and you're studying things and you're not really putting things together, you're having a difficult time or something like that. I am available by phone. Um, I will try to check my email occasionally, but if you really need to talk to me before the exam, then you will have to I suggest please text me. Do not be afraid to text me, okay? Please text me. And if you text me and say, I just need a little bit of clarification, I will get somewhere within a short period of time and I will call you back, okay? So please, I just can't even say please enough to text me, okay? All right, so okay, here are your learning outcomes for the chapter. Please review these because you're gonna know where you need, how you're gonna be expected, what you're gonna be expected to understand here. So please look through your outcomes. Okay, so these next few slides are going to be a little bit of a review for you for A and P. As a matter of fact, these might be a huge review for you. And so okay, so I'm just going to go over them really quick. So you guys know we have genes on our genes. Our genes are on our DNA strand. They give us the information about what specific trait they're talking about. So if your specific trait is hair or your specific trait is a shirt, okay? Now, within that specific trait, we have things called alleles, so a different version of those traits. How many different versions can you have of a trait? How many different versions of a shirt can you have? How many different versions of hair can you have? Okay, those are alleles, all those different versions. So you have dominant and recessive. Dominant is when one allele is expressed over the other, of course. That's why it's dominant. So if you have a dominant and recessive allele, then the trait that's going to be expressed is the dominant, okay? And then recessive alleles, they're on both chromosomes. So you have to have both chromosomes that have recessive alleles on them for that trait to be expressed. Do you understand that? So if you have a short sleeve shirt, I don't know what short sleeve is, or long sleeve. Let's say long sleeve is dominant. So you have to have one chromosome that has a long sleeve allele, and you can have another chromosome that has a long sleeve allele, but if you didn't and you had a chromosome that had a short sleeve allele, because the long sleeve allele is dominant, then you're going to have a long sleeve shirt. Okay? So blonde versus brown. I believe brown is dominant over blonde. Oh, let me go back. So recessive. So in order to have a short sleeve shirt, then each chromosome has to have the short sleeve allele on it to be expressed. Okay? There. And so DNA is the molecule in the cell which stores all the information about an individual, all the unique, everything about that person, okay? Now, chromosomes are the thread-like or X-like structures, shapes. Chromosomes are the little X-like structures of coiled DNA and genes in the nucleus of a molecule. I do know that I feel like you should know that they are in the nucleus. Did I say molecule? I'm sorry. In the nucleus of the cell. You should know that chromosomes and all the information are in the nucleus of the cell. Okay? Oh gosh, got that point across. So these next couple of slides are just everything we just said in that written slide in picture form. So you can look at whichever one you want. Here is yet another one. Okay? So you can look at whichever one you want to help you understand our genes and our alleles. Okay? Here's another that shows you about the dominant versus recessive alleles or genes. Brown up here being brown eye color is dominant. So it's a big B. Little b being the recessive is for blue eyes. Okay. And so this is the mom's eggs. So one of her eggs has the dominant brown eye gene, and one of her eggs has the recessive blue eye gene. Here's the sperm. So one of the sperms has the dominant brown eye gene, and one of the sperms has the the recessive blue eye gene. Now, what are all the different types of combinations you can do, and how do you get brown eyes and blue eyes? Well, in order to get brown eyes, you can have either an ovum, an egg with the brown eye gene and the sperm with the brown eye gene, or you can have the ovum with the this is the sperm with the brown eye gene and the ovum with the blue eye gene. You're going to get a brown eye. This one here is the ovum with the brown eye gene and the sperm with the blue eye gene. You're going to get a brown eye. The only way you're going to get a blue eye is if you get a blue eye sperm and a blue eye egg. Okay? All right. So there's recessive. There's dominant and recessive. I like that little punit square thingy. Okay? Now, chromosomes. So we get half our chromosomes from our mom. We get half our chromosomes from our dad. When those chromosomes are paired up, let's go back. Half our chromosomes, 23 from our mom, half our chromosomes, 23 from dad. When they're paired up, we have 23 matched pairs, which is a total of 46 chromosomes. Okay? 22 of those 23 matched pairs are our body cells. They're the same in the female body and the male body. It's everything except our sex. Basically, all the cells and the genetic material and the genetic information and all the traits about our body are in the 22 autosome chromosomes, and then the last matched pair, the 23rd pair, is the sex chromosome. It determines if we're male or female. Now, females are XX, so they got an X from their mom and an X from their dad. That's a female. A male is a Y chromosome. And so to make a male baby, we have to have the X from our mom. Our mom will always give us an X sex chromosome. Mom will always give us an X sex chromosome. Okay? Dad will either give us a Y male chromosome or an X female chromosome. So whichever one the dad gives us determines which sex we're going to be. Okay? So to write that out, we have 46 chromosome XX, we have a female. Or we have 46 chromosomes XY, we have a male. Okay? All right. Now, when we talk about inheritance, there's a couple of different kinds of inheritance of diseases that we can get. Multifactorial or unifactorial. Multifactorial is 2 or more genes are affected with that disease. It's how a majority of the congenital malformations are formed. So we have that genetic component, or we can have environmental components. Environmental components would be the teratogens. What kind of things are we are getting into our body from the environment that are damaging our genes? Okay, so here are some examples of some of those kind of things. Now, with the inheritance of these, with the multifactorial inheritance, the mildness or the severity depends on how many genes are affected with whatever is going wrong, okay? Unifactorial inheritance is when a single gene for the inheritance is the culprit, okay? So examples of those are autosomal dominant, autosomal recessive, X-linked disorders, and recessive modes of inheritance, okay? So we're going to kind of go over those. Excuse me. I really want you to understand autosomal dominant inheritance. Like, it's really important you should understand this and know this. Okay? So a single gene is altered. And usually we see that in in the family line, in the family tree. Okay? And the parents, so where's my cursor? Oh, here it is. So parents are heterozygous for the trait. That means that they have a healthy gene, and then they have an unhealthy gene, have the trait gene, the disease trait gene. So one of their genes is healthy and one is unhealthy. And in autosomal dominant, we have a 50% chance of passing this to our offspring, okay? And here are some examples of some disorders or diseases that are, oops, did I move? Yes, that are passed in this type of inheritance, autosomal dominant. However, I do not believe no, I'm sorry. However, you do not need to know these specific disorders, okay? Now, autosomal recessive inheritance, each parent has an altered gene, okay? Each parent is heterozygous for the gene, but each parent has an altered gene of that disorder. In other words, dad is heterozygous for the disorder. So he has one healthy gene and one unhealthy gene. Mom, same, okay? Each parent carries that recessive gene. And then, so therefore, they have a 25% risk of passing the trait on to their child. Now, we haven't looked at how we develop eggs and sperms yet, but when we're producing an egg from one single germ cell, okay, we end up with 4 eggs, but that's not true. We end up with 1 egg and 4 polar bodies that disintegrate and go away, okay? So there's 4 chances of that information, that recessive inheritance being, so there's 4 chances of that recessive inheritance being in one of those 4 eggs, but then mom only produces 1, but mom's, I'm sorry, so I'm getting slides ahead, but anyways, so mom and dad, they each, when they're producing a germ cell, they each produce, from one germ cell, they produce 4 individual ovas and individual sperms, okay? So that's why we get the 25%. Gosh, that was terrible. That's why we get the 25% chance of passing it to our offspring, because one of those 4 might have the recessive gene, and one of mom's 4 might have the recessive gene. And if mom's egg that has a recessive gene and dad's egg that has a recessive gene are what's fertilized, then the baby has the disorder. Oh my gosh, that was hard to get out. Now, X-linked dominant inheritance. So the altered gene is on the X chromosome. Which parent gives us the X chromosome? Well, potentially both parents, right? Mom always gives an X chromosome, and dad may give an X chromosome if it's an X sex sperm or if it's a Y sex sperm, okay? So when X-linked dominant inheritance, they only need one of those X chromosomes to have the gene, okay? So remember, dominant inheritance, only one of the genes has to be, or it has to, so this is a dominant, so only one gene has to express it. Remember, like the brown and blue eye color? I'm sorry, came out really difficult. Anyways, so the X-linked dominant in the father, okay? So remember, father gives X sperm if he's making a girl, father gives Y sperm if he's making a boy. So therefore, if this is an X-linked dominant disorder, remember, none of the sons will inherit it. But if it's if the disorder is on the dad's X, then all the daughters will get it because dad gives the sex X to the daughters, okay? Now, If it's the female X-linked dominant disorder, then females have two X chromosomes. They get one from their mom and one from their dad. Um, Therefore, they're twice as likely to get it, for the females to get it than the males, because they have two chances to get it, right? Right? And 50% chance of passing the trait to their child. So if they're getting 50% chance means, they're getting an X from their mom, of course, and then they're getting, if they're a girl, they're getting an X from their dad. And so here's some of the disorders that qualify for X-linked dominant inheritance. X-linked recessive inheritance, they're for some reason more common than X-linked dominant, which I find that confusing. Okay, so maybe one of you smarty pantses can, explain that to me. But anyways, it occurs more frequently in males because they get the single X chromosome from their mom, and if she has the gene, then it can be expressed that way. The father is a carrier because he got an X link from his mom, and so he has a 100% chance of passing it to his daughter, because he's going to give his daughters X's. He's not going to give his daughters Y's. And fathers have a 0% chance of passing it to his sons, because fathers do not give their sons X sex chromosome. They give their sons a Y sex chromosome. Now the female carrier, she has a 50% chance of passing it to her children. Okay? Here are some of those, because moms give X's. That's all she gives. She doesn't give Y's. So if it's on her X, then she could give it to her son, or she could possibly give it to her. She could give it to her daughter, or she could possibly give it to her son. Okay. Anyways, so here is those examples of X-linked recessive inheritance. All right, so that was confusing enough, but please, you kind of need to wait a minute. Let me go back. I think I forgot to point something out. I forgot to point out cystic fibrosis, as you see, is under the examples of autosomal recessive inheritance. That's what I forgot to point out for some reason. All right, cellular division. So this is how we make sperm and eggs. So again, haploid cells are half of the chromosomes, or half of the pair of chromosome. Okay, so that's 23 chromosomes. And we get the other 23 chromosomes, and then they get fertilized or put together, and that makes our 46 chromosomes, which is diploid cells. Okay? Now, when we make gamete cells, we do it through the process of meiosis, and I'm going to explain those to you. The process of mitosis, however, is the process of forming two identical cells from a parent cell. That's how we reproduce our cells in our body. Okay, we regenerate our cells, we reproduce by the process of mitosis. All right, so here is a kind of visual differentiation between mitosis and meiosis. So mitosis, we have our cell, parent cell, and it produces identical daughter cells. Okay. In meiosis, we have our gamete cell, and we have this red or pink color is mom chromosomes. The blue color is dad's chromosomes. They marry up and mix and intertwine and share information, and then they align and then they divide up as chromosomes, and we can see we have some mom, some dad on this one, and then we have dad and some mom on this one. Anyways, they line up, and then they're pulled apart in the cell, okay, or they're pulled to opposite ends of the cell. The cell elongates and then divides, and then we have these two daughter cells, okay, and then they go through basically the same process as here, where now they line up, okay, the chromatids, the spindle fibers, they attach to the chromatids, okay, the little single chromosomes, and then they pull those apart, well, and they pull those to the they the spindle fibers pull these apart, and then they migrate to the different um, regions of the cell, like here, okay? And then this cell elongates just like it did here, and then it divides into two cells. So each one of those does the same process. So we end up with 4 unique cells here. We have 2 identical cells here, okay? 4 unique cells here. So this is meiosis. This is basically how this end product, these 4 little cells here, are sperm. 4 little sperms, okay? Or these are 4 little eggs, except in the female process, it's a little different. From this one cell, the female eggs, she ends up with one cell of 23 chromosomes, and these end up shriveling up and dying, these 3 here. That's because, remember when we talked about last week or whenever it was, how all the fetus, the female fetus, she'll produce all the primary oocytes she'll ever have in her whole entire life. It goes through this process of meiosis, and between the first and second phase, so meiosis is 2 different phases, and it between the first and second phase, it suspends in utero when the baby's born. It suspends in that at the end of that first phase of meiosis, and then that second phase of meiosis does not occur until puberty. She ovulates that one egg, and then that one egg, which would be this one, this would end up to become a polar body. She would ovulate that one egg would be fertilized, and then the last phase of meiosis occurs in that egg, and then the sperm nucleus and then the egg nucleus, they join and create a unique individual. But anyways, so what I just said to you just now, like, it's spelled out. Meiosis phase, meiosis 1 has, well, I've recorded 6 phases. Depending on what source you look at, they break it down in 4 phases, 6 phases, or whatever. But these 2 slides tell you the meiosis 1, what's going on. So that's basically this process right here. Meiosis 1 is this process right here, okay, to produce these 2, okay? And then these 2 goes through this same process to produce these 4, and that's meiosis 2, okay? But anyways, here it is spelled out. And so I'm just going to power through these slides because I want to get to, I don't like reading to you, and you can read this, and you can actually take those slides that have all that written description, and you can split your screen into the written description slides and then these slides that show it in picture form. This is meiosis 1, and this is meiosis 2. And usually I actually draw all this on the board, and I really enjoy doing this lecture because then I can draw all these funky things and you can see my terrible artwork. We have this cell right here. We have these centrioles. So if this is Dawn, if this dude is Dawn, and Dawn is going to produce a bunch of sperm, this is Dawn's parents in here. Dawn's mom and dad. Their genetic traits that they passed on to him, okay? And so anyways, Dawn goes through this, so this is meiosis 1, so he goes through all these phases that are in meiosis 1, okay? These centrioles, part of that is they go to the different, oh, what do they call that? I gotta go back and cheat, hold on. Sorry. That's called the opposite sides. Central migrate to the opposite sides. I thought that was called something. I'm sorry. I'm just a redneck. I don't know these fancy terms. Anyways, so the centrioles, they migrate to the opposite sides of the cell. All these chromosomes have mixed and intertwined and can swapped information, okay? So then we have these chromosomes that are forming, and they have some of Dawn's mom's genetic material, some of Dad's genetic material, Okay, and they're kind of joining together in chromosomes, and then they're lining up in the cell. The centrioles are growing these little spindle fibers. These spindle fibers are now attaching to the different legs of the chromosomes. Okay, and then what happens is they pull them apart. They pull them apart, and then the cell elongates, and then these centrioles pulled this information apart to that side, and this information apart came to this side. They pulled them apart, and the cell elongates and then divides, and then here we have two cells with totally unique information in them. Okay, and then these two cells up from up here, they come down here, and they go through the same process again that happened up here. It's just happening in twice, because we produced two cells, so now it's happening twice with these two cells. Boom, 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 same process as above. Now we've got 4 cells. Oh, wow, that's crazy. So anyways, so we have 4 little sperms here. Now, if this was a female, let's go to this next slide. If you take that picture and do it on a female scale, so these 2 cells right here, okay, let's go back. That's these 2 cells right here at the end of meiosis 1. These 4 cells right here, that's what happened at the end of meiosis 2. These 4 cells here. Okay? Now, that's in the male. So now we've got 4 little sperms. Okay? In the female, from this germ cell, we do all the things that the male did. Okay? Except, okay, so let's go back. So right here, we kind of freeze right in this area. Well, right here, I guess. We kind of freeze. Wait a minute, let me look. Yeah, we freeze right here. Okay. And then puberty, this occurs, okay, in the female, and then she ovulates. Okay, so we've only went through the first meiotic division. Okay, so we only got 2 right here, 2. But in the female, that's all we've done so far. This little guy, he went through this whole process. Okay, now she ovulates. She spits out this one. Okay, this one kind of becomes a polar body. It still goes through the same division like over here, but it's a polar body. It becomes a polar body. This is nothing. This is nothing. This is nothing. Anyways, so we have this one that went through the first meiotic division. The sperm penetrates, and then that initiates the last. Oh, a bug on my computer. Sorry, it scared me. Thought it was a spider. Okay, where was I? Sperm penetrates, and then this cell goes through this last process right here, and she produces this ovum that has been fertilized. This one becomes a polar body and just goes away like these 2 did. Okay, so this is now the fertilized cell. Okay, all right. Now, this is just another slide that kind of shows the same thing, more simplified version. So now we've fertilized, all right? Or, well, not, we haven't fertilized yet. So we have the oocyte and the sperm, they meet in the fallopian tube. When intercourse happens, the man ejaculates over 6 million sperms into the woman. And their flight or their travels up to the fallopian tubes, only 200 of them make it. 200 of the sperm make it, okay? And then, so they get to the egg, and then, you know, all these sperms are just swarming over this egg. There's a chemical reaction called capacitation. That's where the protein coat, this outer coat on top of the sperm head, is removed. And then And then combined with this acrosomal reaction, where the enzyme on the sperm head, it's released and it penetrates the zona pellucida, which is this outer membrane of the oocyte. Okay, so capacitation is when the outer coating of the sperm head is removed, which exposes this enzyme that causes this acrosomal reaction that allows the sperm to penetrate the oocyte. Okay, when that happens, another reaction happens in the zona pellucida um, um, that prevents other sperm from getting in, and that's called polyspermia. So if I was lecturing in front of you, I would ask you if polyspermia occurs, where two sperm penetrate the egg, what kind of pregnancy is that? So that's not going to be on your test because you were tested on that already, but we did cover that. That is a type of pregnancy. It's not a viable pregnancy, but it is a type of pregnancy. So why doesn't one of you impress me and tell me what that is when you see me next week? Okay, after the sperm penetrates the oocyte, the sperm nucleus, the oocyte's nucleus, they fuse and become one, and a single zygote is formed. And that zygote has a diploid number of cells, diploid meaning 2. So a set from mom, a set from dad, 46 total. Okay? Easy peasy. I thought we were going to go fast through this. Now, this I don't know if is a review for you. I would be interested in knowing. I keep meaning to remember if I ask my students or not, but I don't know if you went over this in A&P. So anyways, what happens, so we have the fertilized egg. It goes through this process called cleavage where there's a lot of mitotic division, mitotic division, replication of cells, or dividing of cells, reproducing cells, dividing of cells, reproducing cells, dividing of cells, reproducing of cells. Okay, this is going on. Now, about the third or fourth day, there's about 16-ish number of these cells. And that 16-ish number of cells is it forms a solid ball of cells. It's just a ball of cells, okay? Nothing else but cells. That structure is called a morula, all right? Now, in the morula, as it continues to travel through the fallopian tubes, and starts to enter the uterine cavity, starts to enter the uterus, then fluid is taken into this morula. So all these cells are kind of squished to one side of this ball, and then there's this cavity of fluid, and this cavity of fluid is called a blastocele. Okay, well anyways, blah blah blah, interstitial spaces in the forms of, okay, so when this morula, when the cells went to that, got kind of squished to the one side, those cells are now called the embryoblast, and it's what the the embryo develops from, okay? And so we have that blastocyst, and then we have that fluid-filled space. I'm sorry, we have the embryoblast. I'm sorry. We have the embryoblast, and then we have that cavity of fluid, and then surrounding that is the trophoblast cells. Okay, so let's just look at that. This was really crazy talk. Oops, sorry. All right, so here's your morula. Okay, it's traveling through the fallopian tubes. And then now we're starting to taking on fluid, okay? And so the whole cell now is called a blastocyst. This is the embryoblast part. All these cells squished to this one side, embryoblast, blastocyst, and trophoblast. Okay, and then that's traveling through the uterus. And then that starts to implant into the endometrial layer of the uterus. Remember, perimetrial layer, the outer layer, myometrial, the middle layer, and then the endometrial is the middle layer. Okay, and then the endometrial is the inner layer of the uterus. Okay, so it's embedding into the endometrial layer of the uterus. Boom. All right. So this slide just kind of tells you about that implantation into the endometrium. The cells are continuing to grow. The trophoblast cells, that outer that outer ring of cells, that's what's really kind of invading the endometrial tissue. Capillaries, maternal capillaries, are starting to nourish that embryoblast, that whole ball of cells, that and fluid-filled compartment. This term leukemia, it's just an area in the trophoblast area that's embedding into the endometrium, where it's a space that's developed where the maternal arteries have ruptured, okay, and it develops this space. Well, that space, the leukemia, that's where this intravillous space is created. The intravillous space and the chorionic villi. So those are developing in this leukemia space. This leukemia space was made by these maternal capillaries rupturing. Okay, but this intravillous space and the chorionic villi, that is where the maternal vessels will hook up, and the maternal arteries will give nutrients and oxygen. The maternal veins will pick up all the waste products from the baby is happening in this intravillous space. In the chorionic villi, that's where the capillary terminal end capillaries of the fetal circulation are, and that's where the baby's umbilical vein will eventually, that's where the nutrients are gathered from, this intravillous space dropped off by the mom, and the baby will pick them up in that chorionic villi and then go into the umbilical vein and back to the baby. That's where baby drops off from the umbilical arteries, drops off their waste, and where mom's venous circulation will pick them up in this intravillous space. Okay? All right, so all that's going on. All those vessels are being developed, all those spaces are being developed, and that's what ultimately is going to be happening in that area. Okay, so now the trophoblast, which we said is what forms the placenta that's invading the endometrium. It's still going on by day 25 to 35. Somewhere around 4 months, the placenta is fully developed, and then, but from 4 months on, it's still growing. Like, it doesn't stop growing, but it's by 4 months, it's usually completely developed. So we talked about the maternal blood, the intravillous spaces. We've talked about that. So now the placenta, the functions of the placenta are all these functions listed here. This is how the baby gets their nutrients from these actions here, okay? Gases are exchanged and all that kind of stuff. So a little bit of pathophys right there for you to review. Now here's just a picture of all this kind of stuff. So this is the maternal side. This is the fetal side. We've got maternal arteries and arteries or arterioles. They're coming into this intravillous space. You can see the redness, this intravillous space. The maternal venules, so this is the venous circulation of the mom. This is where mom's picking up all the waste from the baby and going into the mom's venous circulation. Here are the chorionic villi, okay? In those chorionic villi, here we have the arterial and venule, or the terminal ends of the venous and arterial circulation. Here, this is where baby's picking up the nutrients. This is where baby is dropping off their baby's dropping off the waste products and stuff, and then they will end up traveling into the umbilical vein that travels back to the baby. Here's the umbilical arteries traveling to the placenta. Okay, all right. Now, hormones that are important in the pregnancy, are very important for you to know. Okay, just saying. HCG. So it's what we talked about this was it last week? That it maintains that corpus luteum. Remember, when we ovulate and spit out that egg, that follicle that's left in the ovary now becomes the corpus luteum. The corpus luteum produces progesterone and small amounts of estrogen to maintain the pregnancy until the placenta can take over the production of those hormones. But anyways, HCG is what helps maintain the corpus luteum. Okay. And until the placenta is ready to go. Human placental lactogen. So human placental lactogen regulates glucose for the baby. It promotes fetal growth. It also alters maternal protein, carbohydrate, and fat metabolism. So did we go over this? I don't know if we went over this yet. No, we must not have. I don't know. I can't remember. I'm getting so confused. So when a woman becomes pregnant, her protein, carbohydrate, fat metabolism changes in order to accommodate the babies, the growth of the pregnancy, and also keeping mom healthy. All right. Now, progesterone. We're going to talk about progesterone and estrogen, and I want you to think of progesterone and estrogen as yin and yang, because they kind of have some yin and yang type of functions. All right. So progesterone suppresses maternal immunological response to the pregnancy. So we feel progesterone helps her from rejecting the pregnancy, okay, and attacking the pregnancy. Progesterone also decreases myometrial activity and irritability. So if you remember, I told you about my friend who had to have weekly progesterone injections in order to keep her from contracting so preterm and losing her babies. And I had to give her those injections, and they're very painful. Remember I told you that? Yep. So that's what progesterone does. Progesterone constricts myometrial vessels. It decreases maternal sensitivity to carbon dioxide, which is kind of good because she's carrying baby's carbon dioxide as well to excrete it. Inhibits prolactin secretion, relaxes smooth muscle of the GI. So remember, progesterone slows the GI tract to give mom more time to absorb. Increases body temperature, increases maternal sodium and chloride secretion. So estrogen, on the other hand, enhances myometrial activity, promotes myometrial vasodilation. Kind of those things are kind of opposite. Increases maternal sensitivity to carbon dioxide. Softens softens the fibers in the cervix, possibly playing a role in ripening the cervix and preparing it for dilation. Increases pituitary secretion of prolactin to start the lactation process. Increases serum binding of proteins and fibrinogen in preparation for the bleeding that's going to occur with birth to protect mom. Decreases plasma proteins, increases sensitivity of the uterus to progesterone in late pregnancy. Okay, so kind of review those roles of hormones. Very important. Now, development of the embryo and the fetus. So the embryo is what we call this developing thing until about the 8th week, when we call it a fetus. Is it the 8th week? I think so. Yeah, I think it's the 8th week. Sorry. All right. So the yolk sac, very important in nourishment to this growing embryo. It develops about 8 or 9 days after conception. Okay, so it's essential for the transfer of nutrients, I told you. It also is responsible for hematopoiesis, or the development of red blood cells. And then it does that until the fetal liver is developed enough to take over that production. At some point, about 8th or 10th week, I think, it starts to atrophy. Or maybe it's the 12th week. I'm sorry, don't quote me. Atrophies and is incorporated into the umbilical cord. Okay? Now, the umbilical cord, again, is located at the center of the placenta. Hold on. I think, I'm sorry. Hold, please. I can't pause you. Hold on. Call from wireless caller. Call from wireless caller. Okay. I'm sorry. I have my phone in the other room, and I need to know who's calling. So sorry. Okay, hold on. All right. Okay, where was I? Oh shoot. So I'm sorry. Gosh, I'm sorry. Please, bear with me. Hello? No, I'm trying to record a lecture. What do you need? I don't guess so. Like, I don't even know if she's here. The lights are all off in the house. Hold on. Just hold on. I don't... she's not here. Do you need something? Are you okay? I think she's dealing with issues. Okay, yeah, I will have her call you. Okay, thanks, bye. I'm sorry, guys. I can't pause this because if I pause this, then it ends the recording. So I'm sorry you have to put up with this. But life's hard, and I hope you guys forgive me. But anyways, so let me figure out why I'm at the umbilical cord. So we have the embryo, we have the yolk sac, and then we have the stalk that's developing. And the stalk, so when we talked about the placenta and chorionic villi and all these little terminal ends of the fetal circulation, the body stalk, so these blood vessels are starting to form in that body stalk, okay? And again, it connects the embryo to the yolk sac. Remember, I told you the yolk sac gets incorporated into the umbilical cord, so that's kind of how this is being formed. So as these vessels start to develop, they develop into this umbilical cord from the placenta. So we develop into all these little terminal vessels, okay? They start to develop into these cords, or these vessels in the umbilical cord, and we create the 2 arteries and the 1 vein. Remember, the 2 arteries are the vessels that take waste products from the baby to the placenta and drop them off, and mom picks them up. And then the 1 vein is the vessel that picks up the nutrients from mom and takes it back to the baby, okay? That's kind of the not the pathophysiology, the anatomy of what's going on, how that's developing. Anyways, as this cord continues to develop with these vessels in it, then this Wharton jelly is also created. This Wharton jelly, well, it's a connective tissue, but its role is it cushions and protects and surrounds those vessels in the cord. That's what protects them from getting tore, from getting compressed, and all that kind of stuff. All right. Now, maternal and fetal blood. I think I've already touched on this, so I'm just going to drive through this really fast. Maternal arteries, maternal blood flow through her arteries into the placenta through drop off nutrients. Okay, maternal veins that take all the waste away. Fetal umbilical arteries are what drops off the waste into the placenta for the maternal veins to pick up, and then the fetal, I don't know if I just said that right, words are coming out of my mouth. The fetal umbilical vein is what picks up all the nutrients from the maternal artery circulation. Gosh, it's confusing, but it's very simple, so you just got to walk it through your head. All right, where are we now? Okay, so let's see, blood. So we talked about those blood vessels developing. So this is really even more basement anatomy and physiology that's going on. But primarily what we're looking at is, those vessels in the placenta were formed, and then the vessels in that body stalk that become the umbilical cord are developing, and then we start having this primordial heart tube um, that joins those blood vessels and the that connect to that body stalk, that connect to the chorion, that connect to the yolk sac, and this forms the primitive cardiovascular system of the fetus. And then the heart starts to beat by the end of the 3rd week. Okay? So that right there might be something that you might need to know, is that the baby's heart starts to beat by the end of the 3rd week. Okay? And circulate blood, of course. But the 3rd week, after fertilization, the 3rd week should stand out to be important to you, as all the blood vessels have been formed from the placenta through the umbilical cord up through the baby into now this heart, primitive cardiovascular system, and the heart starts to beat. Okay, 3rd week. Boom. All right. Okay. Now, we talked about these 3 unique shunts: the ductus venosus, the foramen ovale, and the ductus arteriosus. The ductus venosus is that channel that connects the umbilical vein to the inferior vena cava. So that umbilical vein is coming from the placenta with oxygen-rich, nutrient-rich blood, and it goes to the liver and passes through this ductus venosus, and then into the inferior vena cava. There is a small amount of blood that goes to the liver, just to supply the liver with a little bit of nutrients. The foramen ovale, you remember, is the opening between the right and left atria. So blood, so let me follow the slide here. Blood empties into the right atrium, to the left atrium, to the left ventricle, to the aorta. Okay, here's the kicker. So blood empties the right atrium from actually the superior vena cava and the inferior vena cava. And it goes from the right atrium straight over to the left atrium, from the left atrium to the left ventricle, into the aorta, and in the fetus, then it goes straight to the aorta, straight to supply the head and the upper extremities. Okay, it doesn't. Yeah, that's right. I don't know what I'm thinking of. Blood, and then blood returning from the head enters the vena cava into the right atrium, and to the right ventricle to the right pulmonary artery. So there's a mixing in the right atrium of superior vena cava blood and inferior vena cava blood. The inferior vena cava blood comes up from the umbilical vein. It's oxygen rich, and it dumps into the right atrium. This low oxygen supply coming down from the superior vena cava mixes in there, or drops in the right atrium. Well, those mix, but this blood, it's got this force pushing down, so it does empty down into the right ventricle. Some of the oxygenated blood dumps down into the right ventricle, but most of that oxygenated blood shoots across through the foramen ovale into the left atrium. Now, if you look at these colors, you can see this mixing. So this is way deoxygenated blood. This is way oxygenated blood. Now look at the right ventricle. The ventricle's got purple blood. If we go down here to the legend, that's medium oxygenated blood. So you can see that mixture, okay, of this hugely oxygenated blood mixing with this hugely deoxygenated blood. Some of it is pushed down into, that mixing is pushed down into here. Okay. And then, so I'm not going to read the slide. I'm going to, the slides that have words on them, I'm going to do this. So then we have our oxygen-rich blood that comes over here, goes down into the left ventricle, shoots up into the aorta, and then goes out these vessels to supply oxygen-rich blood to the upper extremities and the brain and the head and the brain. Okay. And then you can see as it travels through the aorta, now it's getting to be purple blood. So we have this oxygen-rich blood now becoming purple blood. Well, let's look at why that's happening. So this blood that's in the right ventricle, this is mixed blood of oxygenated and deoxygenated blood. It shoots up into the pulmonary artery, okay? And the pulmonary artery typically goes to the lungs to pick up oxygen, but you see how tiny and constricted these vessels look, okay? So blood flow of this mixed blood goes up through the pulmonary artery. It's supposed to go to the lungs to get oxygenated, but it doesn't because this is a fetus. But it goes through this little shunt right here. This is your ductus arteriosus. So we've got mixed oxygenated blood dumping into the aorta with this oxygenated blood that's now supplying the rest of the body. Okay, deoxygenated blood, medium deoxygenated blood, I should say, supplying the rest of the body. This is the whole reason why a fetus, the baby's PaO2 is, the fetus's PaO2 is 40 to 60, whereas an adult PaO2 is 80 to 100. And oxygen saturations for baby in utero can be 60 to 80%. That's normal. That's because of this mixing of blood because of these shunts. Now let's go back here to, where am I at? So in adult person circulation, you know, these pulmonary arteries are very dilated, large amounts of blood. This shunt is closed in the adult person. Large amounts of blood are going in here to pick up oxygen. They come into these pulmonary veins, which are much bigger. These are very constricted in the fetus. And then they're dumping oxygenated blood into the, you know, left ventricle, or left atrium, left ventricle, and out into the body. Okay, that's fetal circulation. Here's that ductus venosus. Oh, I need water. This is that ductus venosus, okay? Excuse me. All right, that's fetal circulation. Boom. Here is it in a ugly diagram type way, if this is easier for you to understand, but I like that picture. Okay, so fetal circulation, we've already talked about all this. So I liked talking when I had the pictures. So here you go. You can look at this, all right? All right, fetal membranes and amniotic fluid. Okay, so let's get there. So there's two, so the bag of waters or the fetal membranes or the amniotic sac, it's actually, those are all terms that you can use for this bag that contains the amniotic fluid in the baby, okay? Those are all the different parts, different names you can hear. But this bag is actually composed of two membranes, two separate membranes, the amnion and the chorion. The amnion is a thinner membrane. It's on the inner, it's on the inside of the bag of waters where the baby is. And then the chorion is the thicker outer membrane. It's tougher. That's the one because it just protects that bag of waters from being broke. And also from the chorion, the placenta is also formed from the chorion, the fetal side. Okay. And remember we talked about the trophoblast, that outer layer of cells with that developing, so that you had the morula, and then you had the the blastocyst, and that can contain the embryoblast and the blastocele, and okay, and then so that blastocyst, the outer membrane of the blastocyst was the trophoblast, okay, and from the trophoblast we have the chorion being developed, the placenta, and the placenta being developed. And within the chorion, we have the amnion, that inner membrane, and then, well, and yeah, the yolk sac, but then the baby, the embryo. Okay. We've already talked about the chorionic villi. Now, those two bag of waters, um, they grow individually, but then they grow together, and then there's this sticky substance that kind of makes them like adhere to one another. But when we're stripping someone's membranes to when they're at term, when we strip their membranes, um, and hopes to put them in labor in the next 12 hours, we're actually rubbing these two membranes together. That's what stripping their membranes is. Okay. Now, the amniotic fluid that's in there, of course, it protects and cushions the baby. It maintains a normal temperature for the baby. It promotes symmetrical growth of the baby. So when the baby's floating in the fluid and not, all parts are able to grow without infringement or impingement or being crushed up against the uterine wall or the amniotic sac wall, baby is freely able to grow normally. Okay. It allows freedom of movement, promotes musculoskeletal development, ensures normal fetal lung maturity. So baby actually does swallow and also inhales amniotic fluid, and it helps with lung maturity, development of lungs. And it moves back and forth across the placental membrane. So, characteristics of amniotic fluid. We start to see it appear about 3 weeks. And then if we were to measure it, like at 10 weeks, so there's about 30 cc's of fluid. So that's only an ounce of fluid. At 24 weeks, there's about 800 cc's and or 800 milliliters. And then, and then should be around 800 or 700 to 800 cc's at birth. So knowing about that much, knowing about 800 is a good number to remember, to keep in mind. I mean, it's pretty common, like if for 10 weeks, it's about 30 cc's, and then it continually adds fluid until about 24 weeks. And then it goes, so almost doubled, like 1.5 times, or 1.5 weeks. What am I saying? Weeks. Shoot, I don't know what I'm saying, but it more than twice the amount of weeks. We've got freaking triplicated, multiplied in several different, up to 800, from 30 to 800 is what I'm trying to say. God, I can't even speak. So anyways, I'm sorry. So fetal urine and fetal lung secretions are the primary mechanisms for the production of amniotic fluid. Okay, so and then, so to control the balance of amniotic fluid or the amount of amniotic fluid, of course, we have urine and lung secretions. And then we also have fetal swallowing, and, they do inhale a little bit, and absorption through the placenta are ways in which baby loses amniotic fluid. So this producing and swallowing and absorbing, should balance out to where baby is still maintaining about 100 cc or 800 cc's of amniotic fluid, after age of viability. Now, when those numbers start dropping, then we're starting to look at oligohydramnios. And when those numbers start increasing, then we're starting to look at hydramnios. Okay. Anyways, amniotic fluid is slightly alkaline. It contains antibacterial properties, protective substances. It contains all other kinds of things: albumin, uric acid, creatinine, all these other words I can't pronounce, bilirubin, vernix, epithelial cells, leukocytes, lanugo. Someone tell me what lanugo is. You guys remember what lanugo is? From the newborn lectures. Anyways, so it contains all those things. All right. Human growth and development. Now, I'm going to spend a little bit of time on the first 8 weeks here and then run through the rest. But all the major organs are formed during the first 8 weeks after conception. That first 8 weeks after conception is termed the embryonic stage, or it's the embryo that's developing, or the embryonic stage. After the 8 weeks, now this embryo is considered a fetus. Hold on. Mother? Mother? Tracy calling something about a ride tomorrow to church and something crazy. Is everything okay when you go? Did you make apologies for me? Like I'm. Yes, I told her that you were doing your 2 lectures in the room. Does she want me to come tonight? Did she say? No, I didn't. Okay. They came and I didn't get that. Okay. Well, I gotta finish. Okay. You know that? No. I'm sorry, guys. Anyways, where was I at? Okay, so when the baby does develop, so it's the cephalocaudal pattern of development, so kind of head to feet type progress of development. So the pre-embryonic period is the first 2 weeks after conception. Now remember, the first 8 weeks is the embryonic period, but the first 2 weeks is the pre-embryonic period. Excuse me. This is where all the, the cellular multiplication and duplication and differentiation are occurring. All the membranes and what's called the germ layers of this embryo, or this the embryoblast cell things, they start producing all these germ layers. And then we already said cephalocaudal development. So we'll talk about those germ layers here in a minute. So let's see. So week 1. Okay, so these are the things that are happening in week 1. I don't expect you to know all these things. Let me just look through and see what I think is really important. You should know that fertilization happens in the fallopian tube. Okay, that the fertilized egg, or the zygote, travels through the fallopian tubes to the uterus. We're having all that mitotic division. We're making that morula. So this, okay, so we're making the morula. Where's my cursor? We're creating a blastocyst. That's where we start taking on that fluid into that ball of cells. And then that blastocyst is covered with the trophoblast cells. And then within the blastocells is that cavity of fluid, which is the blastocele. And then the embryoblast is all the cells that were created, this ball of cells. Okay. Trophoblast invades the uterus, implantation. So just remember, in the first week, the zygote, or the fertilized egg, is traveling through the fallopian tubes to the uterus. We start with the meiotic division of cells, developing cells, and I think the most important thing to know is the trophoblast, which is the outer cells, the part that invades the uterus, implants into the uterus. And that's where the chorion and the placenta are formed. So that's what I want you to know about that. Okay. The second week, trophoblast still invading the uterus. It's dividing in cells. The blastocyst is also invading the uterus. Okay, so week 2, the yolk sac starts to develop. Amniotic fluid cavity appears. Okay, so by the end of the second week, okay, so the second week, things are just continuing. Oh my goodness, what? Everything is just continuing. Um, Cellular division and differentiation and proliferation are just continuing. The important things that I really want you to know about the end of the second week is that the implantation of the blastocyst. So the blastocyst is that whole ball of cell. That whole thing now is completely implanted into the endometrial lining. Okay, we're still having proliferation of cells of the trophoblast and stuff going on, but that blastocyst that contains the embryoblast and the cystocele, that has all is completely implanted in the uterus. Now, this is the end of the second week. This is the point in which now that that ball of cells is implanted in the uterus, now this is when this growing embryo, if exposed to teratogens, can be affected by teratogens. Okay, by the end of this 2 weeks. So that's important for you to know because it's already completely invaded into the uterus. The cells are differentiating, and now there's blood vessels that are starting to be formed. The yolk sac's starting. So this is the start of the period that this fetus or this embryo is susceptible to teratogens after the end of the second week. Okay. All right. Gosh, crap. Dang it. So this is so now we're in the embryonic period, 3rd week to the 8th week. Is the embryonic period. Remember, the first 2 weeks is the pre-embryonic period, but it's kind of all the embryonic period. But anyways, this is where we start having a lot of cellular cell division of that embryoblast, where we're developing these germ layers. From these germ layers are the different parts of the baby are developed, okay? So this is the orgogenic period, or organogenic period, so when the organs are starting to be formed. We have those germ layers. Oh my gosh. Those germ layers are called the ectoderm, the endoderm, and the mesoderm. Okay? You fricker. Sorry. I should just take a second and do this, but anyways, I've wasted enough time. So those 3 germ layers develop into different parts of the baby's development. So you can look at page 202 and it breaks it down. But I broke it down here in the slide. The ectoderm is the outer layer the outer germ layer. It gives rise to skin, teeth, glands of the mouth, but the nervous system. So the big things I like to remember here are skin, teeth, and nervous system. They arise from the ectoderm. That's the outer layer. The inner layer, which is the endoderm, gives rise to the epithelium of the respiratory, GI, and GU tracts. Okay? The mesoderm, this lies between the ectoderm and the endoderm, so I don't know why I've got it listed this way, but anyways it is. Lies between the ectoderm and the endoderm. So this is the middle layer, I guess you could call it. It gives rise to all the connective tissue and the gonads. Okay, so I don't think you need to really, I mean, I would like you to look at these and, kind of have a basic understanding. Try to think of these in terms of things to help you remember, like ecto, being exit or outer, endo being in or inner, and meso being middle. And then trying to put together what do the outer layers, what is the exit? Well, that's our skin, teeth, and nervous system. The middle layer, what does that develop? Well, the connective tissue that keeps everything connected in the middle, I guess, is a good way to think of that. And then the endoderm, the inner layer. So there's your respiratory, GI, and GU tracts. Okay. Oh my goodness, how long is this going to last? All right, so week 4, the germ layers are differentiating into the different organ systems and the different tissues. By the end of this week, we see the 4 limb buds for the arms and legs, and the closed otic vesicles are present. Okay, the nervous system is progressing to develop. Okay, don't really need to focus a lot. Everything's developing. Week 4. Now, oops, shoot. Everything is continuing to differentiate and develop. And then by the end of week 4, we have clear distinctions of the upper and lower limbs. The external genitalias are developed, well developed at this point. And all the major organ systems are developed by the end of this week. Okay? Oh my goodness. All right. So at the end of the 8th week, all of our major organ systems have their basic structure. Okay. Now that's also the end of the embryonic period. The beginning of the fetal period starts at week 9, and this continues until birth or termination of the pregnancy. The baby is considered by worldly standards, by the medical community, as a recognizable human being. The body is continuing to grow. There's still some differentiation of tissues and organ systems. The head growth slows down. Oops, okay, the head growth starts to slow down, and the body really starts to ramp up its growth. So when you see a fetus at 8 weeks, it just looks like they're this big head glob thing and then this little tiny body thing. So the head glob thing starts to slow down and the body glob thing starts to grow more. Now, the last 12 weeks of pregnancy, there's a substantial increase in the size, weight, and length of the baby the last 12 weeks of pregnancy. Okay. The fetal period is less vulnerable than the embryonic period. Remember, because the embryonic period is when all these organs are developing. Okay. Now, if you have an insult during the embryonic period, when organs are trying to develop, then that could really damage the development of an organ. Okay. So that's really serious. Now, teratogens and things like that are bad even in the fetal period, but as this baby ages in weeks gestation, their vulnerability, they get stronger and stronger, and they're able to withstand things. Okay, their organs have been developed. Now the problem is, specialization is occurring and growth. So if baby suffers an insult, that growth is going to be hindered. Okay. So weeks 9 through 10, or 12, I'm sorry. So some of our organ systems are really continuing to start ramping up the functions that they're doing. And you can just read through these next slides and just try to get, a very basic knowledge of what's going on during these weeks. However, please don't spend a lot of time trying to memorize. Okay, so maybe that's a good thing to say. Weeks 13 through 16, here is what's going on there. 17 through 20, and then 21 through 25. So I continue to have these slides, and so I just want you to have a basic understanding and kind of an awareness of what's going on. But please do not try to memorize these slides. You're not going to need to. Okay. Threats to embryonic and fetal development. So of course, genetic factors, maternal exposures to things. We call those teratogens. There's drugs, radiations, infectious agents, environmental things, chromosome abnormalities, medications, and other substances that the woman's using will affect embryonic and fetal development. Torch infections, which we test for prenatally, is your toxoplasmosis. There's a whole list of other infections we kind of group in this other infections category: rubella, cytomegalovirus, herpes simplex virus. So those are broken down in box 7.1 on page 208. So you can just kind of look at what some of those are. Now, things that we know are absolutely teratogens for embryonic and fetal development are fat-soluble vitamins, alcohol, really? Tobacco? I'm shocked. High caffeine intake, cocaine, opiates. I'm being sarcastic. Yes, of course. And so on table 7.4, page 205, drugs or medications and drugs are put into these pregnancy categories. And so I want you to be aware of what those categories are. X is super bad, A is super good, kind of sort of thing. So just kind of look and see what those different categories are. A, B, C, D, X, I think is what they are. Hold on. Okay. Again, we talked about the 2nd to 8th week, the embryonic period being the most vulnerable. Now, we have that initial prenatal exam when a woman comes in, thinks she's, or, confirms she's pregnant. So we do just the same full history and assessment. We do a bunch of prenatal tests. We can educate a woman who's at high risk for genetic disorders about how we can test for those prenatally. Women that are age 35 and older are what we call advanced maternal age, or AMA, and actually the new politically correct terminology is a geriatric maternal pregnant patient, geriatric pregnant patient. But anyways, women over age 35 are at increased risk for having a baby with chromosomal abnormalities. There's trisomies. There's a couple of different types, but it's some sort of combination of, well, let's look at the different ones here. So trisomy 13, trisomy 18, trisomy 21. So it is those chromosomes that are the problem. So they have an extra chromosome. Instead of having the 2 chromosomes, they have the 3 chromosomes on these particular ones. Chromosome 21 is Down syndrome, and that's the most common trisomy problem. Now, when we talk about twinning, we've already covered twins. So we have that monozygotic or identical twin. Monozygotic meaning mono being one, zygotic meaning fertilized egg. So there's those three different kinds of monozygotic twinning, and they are determined by when the division occurs. If it occurs in the first 3 days, or the first 72 hours, then we have 2 embryos, 2 amnions, 2 chorions, 2 placentas. We have 2 placentas, but then the 2 placentas may fuse into one. So that's that dichorion, diamnion, 2 and 2. If the division happens a little later, 4th to the 8th day, we have 2 embryos, 2 amnions, 1 chorion. That is the monochorionic diamniotic twin. And then if the division happens even later, after the 8th day, we have 2 embryos, 1 amnion, 1 chorion. That is the mono or monochorionic monoamniotic twins. This is the most high risk one. The fraternal dizygotic fraternal twins. So monozygotic are more risk than dizygotic, but dizygotic is we have 2 eggs that were each fertilized. We have 2 separate amnions, we have 2 separate chorions, we have 2 separate zygotes or fertilized eggs. We have 2 eggs that got spit out at ovulation, and each one of those eggs got fertilized. Remember, these 2 zygotes are as different as a zygote that was fertilized in 1999 and is as different as a zygote that was fertilized in 2009. Okay? They're 2 whole totally unique individuals. Okay? So minimizing the threats to the baby. We know when we're doing that prenatal visit, we're doing a lot of education with mom. We're asking her about environmental and lifestyle things and letting her know what are risks. We're, of course, supporting her both physically and psychosocially. Now, it's really great if someone comes in for preconception counseling, because if there's any problems with her environmental exposures, with her lifestyle risk factors, if there's any problems with her physical health or with her psychological, psychosocial health, we can do some interventions preconception and get her to the healthiest state she can be before she conceives. So it's super great if people do that, but it's pretty rare that people do that. So anyways, I'm sorry for all the interruptions, and I can't help it. This is just what it is. But I want to go over some things with you, and like I said, it's because I'm terribly sorry that I'm not there with you for lecture. But some of the things I really want you to focus on your studies for the test this weekend is anatomically. I want you to know about the perineum and the true pelvis. I want you to be able to differentiate the different phases in the uterine cycle. I want you to know about hormone production. I'm sorry. I want you to know about the hormones for reproduction. So your follicle stimulating hormone, your luteinizing hormone. Oh, shit. Estrogen and progesterone. I want you to know about hormones in the development of your uterine lining, and hormones for what hormone do you use to test for pregnancy? Gosh, I'm getting so specific. I don't want to be too specific, but I want to be specific enough. Study the information about gender differentiation and identification. Study the section about sex differentiation. Well, I already said that. The role in the external female genitalia development, or what plays in the role of the female external genitalia development. The process of oogenesis. What is that? So study that. Irregular menses. So at the start of puberty, why are young girls' menses irregular? at the start of puberty? What are the physical signs of ovulation? What is cryptorchism? Kidism, sorry. What is that? What does that mean? What do you do? What do you assess for? I want you to know about the Nexplanon and a diaphragm contraceptive options and Depo. Emergency contraception. What contraception would you recommend for a lactating woman? I want you to look at possible sexual dysfunction. I've already pointed this out in this recording. What part of the cell are the genes found in? Understand autosomal dominant inheritance, recessive dominant inheritance, and X-linked dominant inheritance. Understand the process of fertilization. What the yolk sac does. Know about the vessels in the cord and what the purpose of Wharton's jelly is. Understand fetal circulation. Kind of know the general thing about the amount of amniotic fluid after the age of viability. What are the criteria for an abortion? Like, how do we determine what's an abortion? What are the criteria we use to diagnose an abortion? Sorry, I don't know how to say that. Brain development in the fetus. When does that occur? When is the fetus vulnerable or to teratogens? What is just to be able to distinguish between an embryo, the definition of an embryo and a fetus? And I don't know what this says. What? Crap, I'm sorry. I wrote down here what period organs develop. I'm sorry, I don't know what that means, but maybe you better look over what period the organs develop. And I don't mean specifically what organs are developing during week 3 and week 4 and week 5 and week 6 and week 7 and week 8. Okay? Anyways, thank you for putting up with me this week, and good luck on your exam. And please, again, call me if, text me first, and then I will call you if you have any specific questions. Okay? Thank you.